When I introduce myself as a conservation geneticist, a geneticist trying to save rare turtles, usually no clear image appears in people's minds. Do I clone rare turtles to increase their numbers? Actually, rather than replicating one individual, I try to save their genetic diversity. Genetic diversity is important because it helps maintain the health of a population. If the environment changes, a population with higher genetic diversity will be better able to adapt to the new environment. Genetic technologies have been tremendously improved in the last decade. We can now read all the DNA of an organism much cheaper and much faster than before. However, when it comes to utilizing this level of in-depth information to directly address real-world conservation problems, we still lack successful case studies. I have been working on the conservation genomics of a critically endangered Myanmar-specific turtle species, the Burmese roof turtle. Only a handful of individuals were left in the wild, but thanks to successful conservation programs, their numbers had reached hundreds in captivity. My job there was to use cutting-edge DNA technologies to identify, to choose some captive turtles that would be least related to each other and to be reintroduced into the wild, so that their genetic diversity and evolutionary potential would be maximized. On one hand, this was one of the successful case studies actually bridged the gap between the science and real-world applications. On the other hand, there was something missing in the story. Why wasn't it being done by local scientists in Myanmar? Thanks to the support of National Geographic, last year, we ran a project about the democratization of scientific knowledge for biodiversity conservation in Myanmar. We established the first ever molecular laboratory in Hintada University in the south of Myanmar. Here you see me giving hands-on training to the university students on how to use the molecular laboratory tools. We also thought about future generations. We helped these students to meet hundreds of local children in the local secondary schools. They taught them about different types of turtles. They trained them on how to use portable microscope, how to differentiate different types of cells under a microscope, where DNA is located in a cell, and how genetic diversity can help the future of populations. From the secondary school students' point of view, they met their role models, young Myanmar university students, a university professor from their own country, as well as me, a scientist from a different country who works on Myanmar-specific animals. If we would like to address real-world conservation problems more effectively, then amazing advances in scientific knowledge and education should equally be available to everyone, regardless of where they come from. Thank you.